Welcome to the BioInsights, where science comes alive.
I am absolutely thrilled to guide you through today's topic. You know, right now, inside your body, there is a massive, incredibly intricate chemical factory operating at lightning speed. It's building muscle, it's digesting your food, sending signals to your brain, and, well, keeping you alive. But if we zoom all the way in, past the organs, past the tissues, and past the individual cells, we find that this entire biological metropolis is actually constructed from just a handful of foundational elements. So today, we are exploring the molecular architecture of life itself. We're looking at the bricks and the builders.
Now, scientists have discovered tons of different amino acids out in the world, but for human biology, 20 is the magic number. Out of all of them, there are exactly 20 proteogenic amino acids. And proteogenic simply means protein making. These 20 specific molecules act as the fundamental building blocks for literally every single protein in the human body. Think of them as the alphabet of your biology. They combine in endless, unique sequences to write the entire story of you.
Okay, let's dive right into this. Here is our roadmap for today. First up, we'll meet the amino acids. Second, we'll classify these building blocks. Third, we'll look at their powerful derivatives. Fourth, we'll introduce enzyme catalysts. And finally, number 5, we'll look at what happens when this biochemical machinery breaks. Let's get started.
Section 1. Meet the amino acids. Let's transition right into our micro-level focus here. Like I mentioned, you can really think of amino acids as the literal Lego bricks of life. If you wanted to recognize one of these molecular building blocks on the street, you just need to look for 4 things attached to a central carbon, which we call the alpha carbon. So on one side, you've got an amino group. On the other side, a carboxyl group. You throw in a single hydrogen atom just for balance. And finally, the wild card, the unique R group or side chain. This R group is the special sauce. It's what gives each of those 20 amino acids its completely unique chemical personality.
But here's where it gets really interesting. Inside your body, your pH is meticulously regulated, right around a neutral 7. And at this physiological pH, an amino acid acts kind of like a chemical chameleon. The slightly basic amino group actually picks up a proton, giving it a positive charge. Meanwhile, the slightly acidic carboxyl group loses a proton, which gives it a negative charge. It becomes what we call a zwitterion. It has both a positive and a negative pole, but overall, the whole thing balances out to a net neutral charge. It's just a perfect, incredibly delicate equilibrium.
Section 2. Classifying the building blocks. So we have these 20 proteogenic bricks, right? But they definitely aren't all the same. Our bodies actually sort them based on their chemical properties, like whether that wildcard R group is polar, nonpolar, acidic, or basic. And, crucially, we also categorize them based on biological necessity.
Which brings us to a concept you've almost certainly heard about in everyday life: essential versus non-essential amino acids. When you hear the word essential in nutrition, it literally just means it is essential that you get them in your diet, because your body cannot manufacture them from scratch. You have to eat them. Non-essential amino acids, on the flip side, are produced naturally by your own internal machinery. This is exactly why getting complete dietary protein matters so much. You are quite literally importing the critical building materials that your body cannot fabricate on its own.
Now let's look at how we classify them by their chemical charge. Memorizing which amino acids are acidic is super easy. They literally have the word acid right in their names, like aspartic acid. Simple enough. But what about the positively charged, basic amino acids? Well, our sources provided a highly memorable, slightly silly mnemonic for this. Just remember the phrase, her leggings are basic. HLA, histidine, lysine, and arginine. It's just a fun, completely foolproof way to easily recall those basic amino acids.
We also classify these amino acids by how they are broken down in your metabolism. For the purely ketogenic ones, just think of a keto diet. Lamb and liver gives you the double L's for leucine and lysine. For the glucogenic ones, think of sweet things. Honey, mango gives you histidine and valine. And the rest? Well, you can deduce that they fall right into the mixed category. It's a simple, highly structured, and incredibly effective way to keep them all straight.
Section 3. Powerful amino acid derivatives. Okay, let's shift the narrative a bit. We've met the bricks and we've categorized them, but what do they actually do? What do they assemble into once they enter your body's metabolic pathways? Because they definitely don't just sit there. They are transformed into incredibly powerful downstream products that we call derivatives.
And this brilliantly illustrates that exact transformation. Let's look at the assembly of adrenaline. We start with just one single amino acid, phenylalanine. It gets converted to tyrosine, then DOPA, then to dopamine, the neurotransmitter responsible for reward and pleasure. Then it becomes norepinephrine, and finally, epinephrine, which is also known as adrenaline. It's a literal biological assembly line. One single dietary amino acid ultimately generates all of our essential catecholamines that drive your entire fight or flight response. How cool is that?
Let's look at the bigger picture really quickly and see how this plays out across the rest of the amino acids. The downstream importance of having the right amino acids in your system is absolutely massive. Just look at this list. Tryptophan forms niacin, your mood-regulating serotonin, and your sleep-inducing melatonin. Histidine forms histamine for your immune response. Glycine forms porphyrin, which makes the heme in your red blood cells. Glutamate forms the calming neurotransmitter GABA. And arginine? It forms creatine, urea, and nitric oxide. Literally, without these foundational bricks, the chemical communication in your body would simply stop.
So what does this all mean? Here's a really fascinating pivot point. Amino acids by themselves are just passive chemical blocks. So how on earth do they combine to build dynamic machines that can process these metabolic pathways fast enough to actually sustain life?
Section 4. Enzymes, the biological catalysts. Well, the answer to that question is enzymes. When you chain a bunch of amino acids together and fold them up into specific, complex three-dimensional globular proteins, you create an enzyme. These are the ultimate biological machines. They are the builders that take those passive bricks and literally force chemical reactions to happen at the speed of life.
Let's rapidly run through the core traits of how these dynamic machines actually operate. First, enzymes function as biological catalysts, meaning they dramatically speed up chemical reactions. How? By lowering the activation energy barrier that's required for a reaction to even start. They have these highly specific grooves called active sites that bind perfectly to complementary substrate molecules. It's exactly like a lock and key. Best of all, they emerge from the reaction entirely unchanged and unconsumed, ready to go to work all over again. They don't alter the overall free energy or the delta G of the reaction. And because their shape relies on those delicate amino acid bonds we talked about, they are highly sensitive to shifts in temperature and pH.
Now, if all of that sounds just a bit too abstract, I absolutely love this analogy from our source material. Imagine you are traveling from California to New York. You could drive a car over the mountains, that's going steeply uphill, it takes a massive amount of time, and you're burning a ton of energy. Or you could take an airplane. The airplane is the enzyme. It's the ultimate shortcut. It uses far less activation energy to get you there much faster. But, and this is absolutely vital to understand, whether you take the car or the airplane, you still end up in New York, right? The enzyme doesn't change your final destination or the chemical equilibrium. It just gives you a faster, lower energy pathway to get there.
So the crucial point here is that these enzymatic airplanes are highly specialized tools, and they are deployed exactly where they are needed. For instance, we have intracellular enzymes, like catalase, which works entirely inside your cells to rapidly break down toxic hydrogen peroxide into harmless water and oxygen. And on the flip side, we have extracellular enzymes, like amylase and trypsin. These are produced by the pancreas but are actually sent outside the cells into the digestive system to break down huge, bulky starches and proteins into manageable sugars and individual amino acids.
Section 5. When biochemical machinery breaks. Okay, so we've seen the sheer perfection of this system, right? The bricks building the enzymes and the enzymes processing the bricks. But what happens when there is a microscopic flaw in the blueprint? We need to ground this chemistry in real-world clinical reality. Because when this biochemical machinery breaks down, the consequences for human life are really profound.
Take Hartnup disease, for example. This is an autosomal recessive condition where a person's kidneys simply fail to reabsorb neutral amino acids, primarily tryptophan. It literally just slips away in the urine. And because of that one single failure, the body doesn't have tryptophan. Now, as we saw earlier, without tryptophan, you cannot form niacin. And a niacin deficiency leads directly to pellagra, which is a devastating systemic condition characterized by the 3 Ds: severe diarrhea, dementia, and dermatitis. It's a massive whole-body domino effect caused by the missing pathway for just one single amino acid.
Or consider what happens when a builder actually goes missing. The branched-chain amino acids we talked about, isoleucine, leucine, and valine, require a very specific enzyme to be broken down. If a patient is genetically missing that enzyme, which is called branched-chain alpha-ketoacid dehydrogenase, those amino acids build up to incredibly toxic levels. This results in maple syrup urine disease. The mnemonic to remember this one is, I love Vermont maple syrup, can I have a sip? ILV stands for isoleucine, leucine, and valine. And SIP stands for the tragic hallmark symptoms of the disease: syrup-smelling urine,
intellectual disability, and poor oral intake. Again, the absence of just one enzyme changes everything.
Finally, let's look at sickle cell disease. Hemoglobin, which is the massive protein that carries oxygen in your red blood cells, is made up of hundreds of amino acids. In a healthy adult, hemoglobin A features the acidic polar amino acid glutamic acid, right at position number 6. But in sickle cell disease, a single point mutation swaps out that glutamic acid for valine, which is nonpolar. Think about that. Literally just one swapped Lego brick out of hundreds. But that single tiny swap completely alters the protein's entire shape under low oxygen, causing the whole red blood cell to sickle. This leads to severe pain, hemolysis, and extensive organ damage.
It is honestly truly staggering when you take a step back and look at it all. We are these vast, breathing, thinking beings, but our entire existence rests on the precise microscopic alignment of atoms. If a single amino acid swap or one single missing enzyme can completely alter the trajectory of a human life, just pause for a second and ask yourself, what millions of perfectly choreographed, completely untracked biochemical reactions are happening inside of you at this exact moment just to let you sit there and watch this explainer? It is a delicate, wildly beautiful chemical balance. Thank you so much for joining me today, and keep exploring.